Hi students, Doc Ian here. This is the continuation of our lecture on the nucleus. This lecture is intended to discuss meiosis. But before we'll discuss meiosis, let us first differentiate it from mitosis. Mitosis is the cell division of somatic cells. Somatic cells are diploid, meaning they have a total of 46 chromosomes. A parent cell will produce two daughter cells in mitosis. Both of these daughter cells will have 46 chromosomes each. Meiosis, on the other hand, is the cellular division of the germ cells, which include the egg cell and sperm cell. We will try to figure out later how many daughter cells will be produced in meiosis and if the daughter cells will be haploid or diploid. Again, meiosis is the form of cell division involving the gametes or the germ cells, the sperm and the egg, wherein the number of chromosomes will be reduced. So expect that the daughter cells of meiosis will have half of the total number of chromosomes, and they will be referred to or described as haploid. Please take note that in meiosis, the cell has to undergo two cell divisions. That's why we have meiosis I and we have meiosis II. Let's start with meiosis I. Whatever stages we have discussed in prophase will be the same stages that we will cover in meiosis I. Expect that before the cell will enter the prophase I of meiosis I, there should be an interphase. And you know very well that one of the stages in interphase is synthetic phase. So expect that the number of chromatids will be doubled. The precursor cell for the mature sperm cells is the spermatogonium. Please take note that the spermatogonium has 46 chromosomes. So expect that the spermatogonium will initially have this set of chromosomes. And since meiosis I will start with interphase, expect that the chromosomes in the spermatogonium during the meiosis I stage until the metaphase I will all be looking like this. Count the number of centromeres. You have a total of 46. So that will give you a total number of 46 chromosomes. Count the number of arms. So for each centromere, you have 2 arms. So that will give you a total of 92 chromatids. Meiosis I and meiosis II have the same stages similar to mitosis. It's just that we need to indicate number 1 or number 2 in the stages of meiosis I and meiosis II so that we will be guided if the cell is in the prophase of meiosis I or meiosis II. So therefore, the 4 phases in meiosis I are the following: prophase I, metaphase I, Anaphase I, and Telophase I. Whatever are the events that you have memorized for the prophase, metaphase, anaphase, and telophase of mitosis may also be the same with the events that will occur in the prophase I, metaphase I, anaphase I, and telophase I of meiosis I. It's just that there are slight differences. Just like in the prophase of mitosis, in prophase I, the chromosomes will condense, the centrioles will duplicate, and they will start migrating towards the opposite poles of the cell. Then you will have the synthesis of the mitotic spindle. Expect that the nuclear envelope and the nucleolus will disappear. But prophase I is the longest stages in both meiosis I and meiosis II. Why? Prophase I is the longest among all the stages in meiosis. It's because in prophase I, the cell has to undergo several stages. And for you to easily remember the correct sequences of these stages, you have to sing with me. Lizapadidia, Lizapadidia. Le, leptotin, and it will be followed by zygotin. Pa, pakitin. D, diplotin, and then diakinesis. That's why, as shown in the picture, what will follow after diakinesis, which is the final stage of prophase 1, is metaphase 1. The next thing that we will do is to discuss the events or the changes that will happen in the chromosomes in leptotin, zygotin, pakitin, diplotin, and diakinesis. Leptotin is the earliest stage of prophase 1. So expect it is in this stage where the condensation of chromatin to form the chromosome will happen. The important thing I want you to remember about leptotin is that it is also in this stage where the homologous chromosomes will search for each other. Remember, for each chromosome, we have one that was inherited from mother and one that was inherited from the father. So say, for example, chromosome number 8. You have chromosome 8 from the mother, chromosome 8 from the father. So what will happen in leptotin is the chromosome 8 from the mother will look for the chromosome 8 from the father. So they will now pair as homologous chromosomes. That's why we call that process as homology search, and that is happening in leptotin. So for you to remember that there is homology searching in leptotin, you have to remember no one is left alone because they will be paired. Take a look at these two chromosomes in the picture. Appreciate that they are already duplicated because before the cell will enter prophase 1, the cell has undergone interphase, particularly the synthetic phase. So let's say, for example, this is chromosome number 8. So the red-colored chromosome is the chromosome number 8 from the mother, and the green-colored chromosome is chromosome 8 from the father. They will now undergo the second stage of prophase 1, and that is zygotin. The important event in zygotin is that these two homologous chromosomes will form synaptotemal complex, the one that is labeled in the picture. And this synaptotemal complex will always cause these chromosomes to be bound together. I want you to be reminded that in mitosis, the chromosomes will all align in a single file at the equatorial plate or center of the cell, so that if the spindle fibers will pull them towards the opposite pole, you can see that the chromosomes will be torn into two, and you will now have the sister chromatids migrating towards the opposite poles. In the case of meiosis, homologous chromosomes pair, and they are bound together because they will form the synaptotemal complex. That's why they cannot assume the single file arrangement in metaphase 1. Instead, when they are already in metaphase 1, these chromosomes will be at the center of the cell as pairs. So what will happen is, these chromosomes now, once they will be pulled by the mitotic spindles, they will be separated as one whole chromosome, and that one whole chromosome will be pulled towards the opposite side of the cell. So I want you to take a look at the chromosome that is encircled right now. So let's say that's chromosome number 8, and that was inherited from the mother. So the blue-colored chromosome beside it is the chromosome number 8 that was inherited from the father. So what happens in meiosis 1 is that the whole chromosome number 8 that was inherited from the mother will be pulled to the right side. And it is only the daughter cell on the right side that gets the chromosome number 8 from the mother. So the cell on the left side will only get the whole or chromosome number 8 from the father. And we cannot afford to let that happen, because the mother and the father should have something to say about the traits of the daughter cells, because we're talking about sperm cells and egg cells and meiosis. how to solve this problem? Na when we will pull the chromosomes towards the opposite side, somehow the chromosome 8 inherited from the mother will carry something from chromosome 8 that was inherited from father, so that the daughter cell on the right side of the screen will have a chance of getting the traits from the chromosome number 8 that was inherited from the father. That will now be answered by the third stage of prophase 1, and that's pakitin. In pakitin, there is crossing over of chromosome pairs. I want you to take a look at the blue and the red colored chromosomes. So let's say, for example, these are chromosome number 8. Blue was inherited from father, red was inherited from mother. Since they are already in pair after zygote, so because of the formation of the synaptotemal complex, what will happen is, in pakitin, they will try to exchange some portions of their structures. That's why after the completion of the pakitin, you can see that the chromosome number 8 that was inherited from the father will have a small portion derived from the chromosome number 8 that was inherited from the mother. So if these chromosomes will be pulled towards the opposite sides, the daughter cells will have a chance of getting the traits from the other homologous or pair of chromosomes. Chiasma or chiasmata is the portion in the homologous chromosomes where the exchange of genetic material occurred. So take a look at this picture. You have here an area that is labeled as chiasmata. So this is the site for the crossing over of genetic materials. Pakitin will be followed by diplotin. In diplotin, the crossing over has already been completed, and there would be further condensation of the chromosome. That's why they will appear to become thicker and shorten. This time, the synaptotemal complex will start to break down, so that these chromosomes will now be pulled towards the opposite sides later on in anaphase. The condensation of the chromosomes, making them appear thicker and shorten, continuous with diakinesis. The important events happening in diakinesis is the other events that we usually associate with prophase. We have the disappearance of the nucleus and the dissolution of the nuclear envelope, and the migration of the duplicated centrioles towards the opposite poles of the cell, and the synthesis of the mitotic spindle. Take note, we are still in the prophase 1, so expect that these events will really occur. As mentioned a while ago, in Metaphase 1, the chromosomes will align at the center of the cell by pair because of what we call as the homologous pairing or homologous searching that happened during the start of prophase 1, and that is in leptotin. And these chromosomes cannot assume the single file arrangement because they have the synaptotemal complex that was formed during the zygote stage. The breakdown of the synaptotemal complex will now allow the homologous chromosomes to be separated from each other. That's why, as the cell will enter the anaphase stage, the homologous chromosomes will be pulled towards the opposite poles or opposite sides. But I want you to appreciate in the picture that one of the chromosome bears a small portion that was taken from its pair. It's because of the crossing over that happened in the pakitin stage of prophase 1. In anaphase 1, the homologous chromosomes separate and move towards the opposite poles. Sister chromatids on the same chromosome remain attached to each other because you are going to pull one whole chromosome. So the sister chromatids will never separate from each other, which is very different from the anaphase of mitosis. Because in the anaphase of mitosis, sister chromatids are pulled towards the opposite side. This time, the sister chromatids will move towards the opposite sides together. The events in telophase I are basically the same as that of the telophase of mitosis. The chromosomes will become less condensed, the nucleolus and the nuclear membrane will start to reappear, and then the mitotic spindles will disappear, and then this telophase I will be followed by the separation of the daughter cells in the process called cytokinesis. The spermatogonium and the oogonium have this set of chromosomes prior to the interphase. But because of the synthetic phase of the interphase, their chromosomes will look like this one. They have a total of 46 chromosomes with 92 chromatids. And then the chromosomes, focus on chromosome number 8, the chromosomes will align by pair at the center of the cell at metaphase I. And these chromosomes will be pulled towards the opposite sides with the sister chromatids moving together towards the same side of the cell. So, expect that one of the daughter cells will have this set of chromosomes. So I want you to take a look at chromosome number 8. So this could be the chromosome that was inherited from the mother. But I want you to imagine that it will have a small portion of it that was derived from the other pair, the chromosome number 8 that was inherited from the father. And that chromosome now is present on the other cell. At this point, I want you to count the number of centromeres. You can only count 23 of them. So meaning, there is the reduction in the number of chromosomes in meiosis I. That's why meiosis I is also known as reductional cellular division, because there is reduction in the number of chromosomes. So let's have a review. Spermatogonium and oogonium originally have 46 chromosomes with 46 chromatids. After going through the synthetic phase of interphase, the chromosome number will still be the same, 46, because we only base it on the number of centromeres, but the number of chromatids will become 92. After the completion of the meiosis I, the chromosome number now will be reduced to half. And then, same as the number of chromatids, from 92, it will be reduced to 46. Shown in this photo are the different stages of meiosis I and meiosis II. What I want you to take note is that there is no interphase prior to the start of meiosis II. Since there is no interphase prior to meiosis II, so there would be no duplication of DNA and chromatids in meiosis II. So expect that the daughter cells of meiosis I will enter meiosis II, each having 23 chromosomes and 46 chromatids. Now, the events that will happen in Prophase II, Metaphase II, Anaphase II, and Telophase II are similar to that of mitosis. So expect in Prophase II, you have the further condensation of the chromosomes, the duplication of the centrioles, and their migration towards the opposite poles of the cell, with corresponding synthesis of mitotic spindle. You have the disappearance of the nucleolus and the nuclear membrane. In metaphase II now, the chromosomes will be found at the center of the cell in a single file arrangement. This is just to review you of the difference in metaphase I and metaphase II. In metaphase I, the homologous chromosomes are aligned at the center of the cell by pair because you have the synaptotemal complex. While in metaphase II, the chromosomes will be aligned in the center of the cell in a single file arrangement. So expect that in anaphase II, the chromosomes now will be separated into two sister chromatids similar to anaphase of mitosis, and these sister chromatids will travel towards the opposite pole. So the events in telophase II are similar to that of telophase of mitosis. You have the reappearance of the nucleus and the nuclear membrane. You have the chromosomes becoming less condensed. The cell will elongate to allow the cytokinesis to occur after telophase II. Meiosis I resulted to the production of two daughter cells, each having 23 chromosomes or 46 chromatids. In meiosis II, the two daughter cells will give rise to four daughter cells. And we will try to identify how many chromosomes are present in each of the four daughter cells. Please recall that this is the chromosomal makeup of one of the daughter cells in meiosis I. As you can see, you only have there 23 chromosomes, but you have 46 chromatids. Recall that in Anaphase II, the sister chromatids will be separated from each other. So take a look at chromosome number 8. So in Anaphase II, we will separate the two chromatids. If they are separated, they will now be considered as separate chromosomes. But then again, each of these sister chromatids will go to the daughter cells. So each of the daughter cell now will have still 23 chromosomes, but this time they only have 23 chromatids each. And since we have two daughter cells from meiosis I entering meiosis II, each of them will give rise to their own respective daughter cells. So there would be how many? 4 daughter cells with 23 chromosomes each. And this will now be the chromosomal makeup of one of the 4 daughter cells in meiosis. As you can see, for each chromosome number, you only have 1 chromosome and 1 chromatid. Since in meiosis II, you only separated the two sister chromatids without reducing the number of chromosomes, therefore meiosis II is referred to as equational cell division, while meiosis I is named or also known as reductional cell division. We will now highlight the differences between meiosis and mitosis. Homologous chromosomes pair up in meiosis because they are bound by the synaptotemal complex formed during the zygote stage of prophase I, while chromosomes do not pair up in mitosis. That's why they can be arranged in single file arrangement in metaphase. Crossing over is possible with meiosis, particularly at the packeting stage of prophase I, and there is no crossing over in mitosis. The germ cell, the sperm, and the egg has to undergo 2 cell divisions in meiosis. You have meiosis I and meiosis II, while somatic cells only have to undergo 1 cell division. There are 4 daughter cells produced in meiosis, and each of the daughter cell only has what? 23 chromosomes. So you have 4 haploid daughter cells, while you only have 2 daughter cells in mitosis, and each of them are diploid. There are 2 types of cell death. We have necrosis and we have apoptosis, and we will discuss them one by one. Necrosis is a type of cell death that results from an injury. So expect that the cells in necrosis are exposed to unfavorable physical or chemical environment. So that could be brought about by sudden change in the pH becoming more acidic, causing the cells to die, sudden change in the temperature, or if the cells will be deprived of oxygen, just like in the case of acute myocardial infarction or heart attack, the cardiac muscles will die, causing now the dysfunction in the heart, causing now the problem with pumping the blood all throughout the body for the organs to receive oxygen. Necrosis can also be because of exposure to radiation, trauma, and it can be caused by microorganisms in the form of viruses, bacteria, and fungi, and as well as parasites. So what are now the features of necrosis? Since there is the presence of an injurious stimulus, the injurious stimulus will cause damage to the cell membrane. You know very well that the cell membrane selects the substances that can go in and out of the cell. If that cell membrane is damaged, ions and as well as water are allowed to go inside the cell. If there is accumulation of electrolytes or ions and water inside the cell, that will now cause the cell to swell. Further swelling of the cell will eventually cause it to undergo lysis or bursting. Another unique characteristic of necrosis is that the DNA will be randomly fragmented by the injurious stimulus. Apoptosis, on the other hand, is a type of cell death that does not result from the presence of injurious stimuli. This is actually the mechanism of the body in eliminating cells that are no longer needed or cells that have the potential to become cancer cells. So if the cell will detect that there's something wrong inside it, it will activate a certain mechanism which will cause that cell to die. Now, in apoptosis, there is internal autodigestion, but the cell membrane of the cell remains intact. That's why you expect that the cells in apoptosis will never swell and burst. So these are the features of apoptosis. As mentioned a while ago, once the cell will undergo apoptosis, it will activate some mechanisms inside it to facilitate its death. One of the mechanisms is the activation of endonuclease, and this endonuclease will cleave the DNA of that cell at specific sites. So remember, in necrosis, the DNA is randomly fragmented. In the case of apoptosis, the endonuclease will cleave the DNA at specific sites. One of the mechanisms that will also be activated in apoptosis is the organization of the cytoskeleton or the filaments inside the cell. So, shown in the picture, you have here the microtubules, the actin filaments, and the intermediate filaments. So, these filaments will interact with each other. Since they will be pulling each other, they will now cause the cell to shrink in size. That is in contrast to the swelling of the cell in necrosis. Another unique characteristic of apoptosis is that the cytoskeleton, the different filaments that we have inside our cell, will also organize and cause the membrane to form blebs. That's why there is membrane blebbing in apoptosis. So take a look at this cell. This cell is about to undergo apoptosis. So take a look at the significant decrease in the size of the cell brought about by the interactions of the different filaments or the members of the cytoskeleton of the cell. The filaments or the cytoskeleton will also cause the plasma membrane of the cells to undergo blebbing. And this blebbing will eventually lead to the formation of the apoptotic bodies. And these apoptotic bodies now will be removed from the body by the action of the phagocytes. What I like about apoptosis is the apoptotic bodies will not allow the contents of the cell to leak out because there are chemicals inside the cell that can induce inflammation. And since apoptosis is a physiologic process, it's a normal process, it's not supposed to induce inflammatory reaction, the cell must not allow its content to leak out. That's why you have there the formation of apoptotic bodies. Going back to necrosis, you have there the presence of the injurious stimuli. Expect that there is already inflammation. What will make the inflammation worse is the release of the chemicals from the cytoplasm of the cell because there is cell swelling which will eventually lead to the cell bursting or undergoing lysis. Another unique characteristic of apoptosis is that there is the loss of mitochondrial function. The membrane of the mitochondrion will become more permeable or leaky, allowing now the escape of cytochrome C. And this cytochrome C will activate caspase enzyme. So take a look at the picture of the mitochondrion in the slide. You have there the release of cytochrome C from the mitochondrion because of the increased permeability of its membrane, and that cytochrome C will activate an enzyme which is called caspase, and caspase now will cause the degeneration of the organelles of the cell because the cell is about to die. So at this point, I want you to differentiate necrosis and apoptosis based on the parameters mentioned in that table. Cell swelling is seen in necrosis, brought about by the cell membrane damage, while cell shrinking is evident in apoptosis because of the organization of the cytoskeleton or the filaments within the cell. Now, which among them is characterized by the damage to the plasma membrane brought about by the injurious stimuli? Yeah, that's necrosis. Which among them is characterized by the formation of blebbing in the plasma membrane? That is the apoptosis, leading to what? The formation of apoptotic bodies later on. In terms of the fragmentation of the DNA, which among them is characterized by random DNA fragmentation? That's necrosis. Specific DNA fragmentation? Yes, that's apoptosis. Which among them can be caused by lack of energy or ATP and lack of oxygen or hypoxemia? Yeah, that's necrosis. And which among them involves the action or activation of the enzyme caspase? Apoptosis. Please remember the leaking of cytochrome C from the mitochondrion, activating now the caspase enzyme, which will cause the degeneration of the organelles of the cell. And these are now the correct answers. Thank you for listening to my lecture on Nucleus. Good luck with our quiz.